Welcome back to BioB 160 Principles of Living Systems. I'm Ruth Reitzman, your instructor for the course, and this episode will look at the chromosomal basis of inheritance. Let's get started. Mendel's work on the inheritance of traits in pea plants showed that physical traits were passed from one generation to the next through particulate heritable factors, but Mendel had no idea what those factors were and where they were located in the cell. However, in the late 1800s, studies on the cell's nucleus showed that these factors, which we now call genes, are physically located on the chromosomes in a cell's nucleus, and this led to the development of the chromosomal theory of inheritance. The chromosomal theory allows us to understand the behavior of chromosomes during meiosis and explains Mendel's laws of segregation and the law of independent assortment. If we examine the chromosomes in Mendel's pea plants, we can track how the physical characteristics were passed from one generation to the next. In the case of the dihybrid cross between true breeding parental peas with yellow-brown seeds and peas with green wrinkled seeds, we can see that the cells have the traits for seed color and seed shape on separate chromosomes. Since each parent is true breeding, meaning they are homozygous for both traits, they will produce only one type of gamete. When these gametes fertilize, the F1 progeny are yellow and round and are heterozygous for both the seed color and seed shape. When the F1 plants produce gametes, the laws of segregation and independent assortment predict that there are 4 possible types of gametes that will be produced, and these 4 types are produced in equal frequency. As a result, 1/4 of the gametes contain both dominant traits, 1/4 gametes contain the dominant yellow trait with the recessive wrinkle trait, 1/4 of the gametes contain recessive green with dominant round, and 1/4 contain both recessive traits. As we saw in the last episode, when these 4 types of gametes are fertilized with the 4 types of gametes from another F1 plant, the result are 16 possible genotypes. However, when we look at the phenotypic categories, we can see that the 16 genotypes can be combined into 4 phenotypes, giving us plants with 9/16 round yellow seeds, 3/16 green round seeds, 3/16 yellowed wrinkled seeds, and 1/16 green wrinkled seeds. Although plants provide a great model for studying inheritance, they have some disadvantages. They have long generation times, and until greenhouses are developed, they can only be cultivated during the growing season. In the early 1900s, the geneticist Thomas Hunt Morgan began studying the genetics of the fruit fly, Drosophila melanogaster. Drosophila provides some advantages as a genetic model organism since it produces a complete generation in 2 weeks, and it produces many offspring. It also has a small chromosome number, just 4 pairs. This photo from around 1910 shows Thomas Hunt Morgan in his lab at Columbia University. T.H. Morgan's careful examination of thousands of fruit flies led to the discovery of a fly with white eyes rather than the typical red eyes. Morgan then wanted to see how the white-eyed trait was inherited, and to do this he set up a series of crosses. Morgan set up an experiment in which he mated a white-eyed male fly with a red-eyed female fly. In the F1 generation, all of the offspring had red eyes. To follow the inheritance of the trait, he then mated the F1 flies and observed the eye color of the F2 generation. Morgan assumed, since he saw F1s with red eyes, that the red-eyed color was dominant over white eyes, and that he predicted if he crossed the F1s, he would see a 3-to-1 ratio of red eyes to white eyes in the F2 generation. When he examined the results of the F2 generation, he did see a 3-to-1 ratio of red eyes to white eyes. However, he noticed something very interesting and unpredicted. All of the female flies had red eyes. However, the male flies were 50-50, red eyes to white eyes. Morgan had found something that Mendel had not observed, a gene that was linked to the sex of the individual. The white-eye gene is a sex-linked trait. How can this be related to chromosomes and meiosis? This diagram shows the chromosomes of a true-breeding red-eyed female and a true-breeding white-eyed male. In Drosophila genetics, the mutant trait is given a lowercase letter. For white eyes, this would be the lowercase w. The normal or wild-type trait is indicated with a superscript plus. So for red eye color, the trait is represented by W+. Sex determination in fruit flies is similar to humans. Individuals with 2 X chromosomes are females, and individuals with 1 X and 1 Y chromosome are males. The Y chromosome is a very small chromosome and carries very little genetic information except for the genetic signals that determine maleness. The crosses set up by Morgan started with a red-eyed female indicated as a W+, and a white-eyed male indicated with a W. Since the eye color trait is carried on the X chromosome, females will have 2 copies of the gene, but males will have only 1 copy since they have a single X chromosome. If we look at the Punnett square for the parental cross, we can see that the parental red-eyed females produce gametes with X chromosomes, and each X chromosome carries a W+ gene. The parental white-eyed males produce 2 types of gametes, one half with X chromosomes and one half with Y chromosomes. The X chromosome carries the W white-eye gene, and the Y chromosome lacks any eye color traits. The F1s are all red-eyed, however, the females are heterozygous, while the males only can carry 1 eye color trait. Since males inherit their X chromosome from moms, all of the males have a W+ or red eye color gene. Now let's look at what happens when we cross the F1s. The F1 females are heterozygous, so one half of their gametes contain X chromosomes carrying the wild-type W+ gene, and one half carry the W white gene. The males will also produce 2 types of gametes, one half with the X chromosome carrying the wild-type gene, W+, and one half with the Y chromosome which lacks the eye color gene. We can use the Punnett square to predict the genotypes of the offspring in the F2 generation. All of the F2 females will have red eyes, but one half of the females will be homozygous for the W+ gene and one half will be heterozygous. For the males, one half will be red-eyed and one half will be white-eyed. The Y chromosome inherited from their father will not contribute to the eye color trait, but the X chromosome from their mother will determine their eye color. Since the mother was heterozygous, one half of the males are red-eyed and one half are white-eyed. Now let's examine chromosomes and sex determination in humans. Humans, like fruit flies, have X and Y chromosomes. Females have 2 X chromosomes and males have 1 X and 1 Y chromosome. As with fruit flies, the father's sperm determines the sex of the offspring. If the sperm carries an X chromosome, the resulting child will be female. If the sperm carries a Y chromosome, the resulting child will be male. The Y chromosome is much smaller than the X chromosome, and it carries a few traits, mostly genes related to the development of male sex characteristics. The other chromosomes are called autosomes. So the human genome consists of 46 chromosomes. 44 are autosomes, and the other 2 chromosomes are the sex chromosomes. Now let's look at how sex-linked traits are inherited in humans. Some examples of sex-linked traits are red-green color blindness, hemophilia, a blood clotting disorder, and muscular dystrophy, a disease that causes muscle deterioration. Let's consider the inheritance of one of these sex-linked traits, red-green color blindness. The first case to consider is a normal female and a colorblind male. In this example, the capital N refers to normal vision and the lowercase refers to red-green color blindness. If a woman is homozygous for normal color vision, all of her eggs will carry X chromosomes with normal color vision genes. A male with color blindness will produce gametes with X chromosomes carrying the color blindness gene and Y chromosomes which lacks genes for color vision. In the resulting offspring, both male and female will have normal vision. However, the females will inherit an X chromosome from their dad that carries the color blindness gene. So the females are called carriers since they carry a defective gene even if their color vision is normal. The second case to consider is a carrier female and a normal male. If a woman is heterozygous for color blindness, one half of her eggs will contain X chromosomes with normal color vision genes, and one half will contain X chromosomes with the color blindness gene. A male with normal vision will produce X chromosomes carrying normal color vision genes and Y chromosomes. If we look at the Punnett square, we can see that all of the females will have normal vision, but one half of them will be carriers of the color blindness gene. The boys in this case will have a 50-50 chance of having color blindness. One half of the boys will get an X chromosome with the color blindness gene, and one half will have normal color vision. One thing to note about the inheritance of sex-linked traits is that men cannot be heterozygous. They either have the defective gene or they have the normal gene. The term hemizygous is sometimes used to describe the genes carried by a male X chromosome. The third case to consider is a carrier female and a colorblind male. As we saw in the last case, if a woman is heterozygous for color blindness, one half of her eggs will contain X chromosomes with normal color vision genes, and one half will contain X chromosomes with the color blindness gene. A colorblind male will produce X chromosomes carrying the colorblind gene and Y chromosomes which lack genes for color vision. If we look at the Punnett square, we can see that one half of the females will be carriers with normal color vision, and one half of the females will be colorblind. For males, one half will have normal color vision and one half will be colorblind. If you study these three cases, you'll see that you can apply them to any sex-linked trait. The key to working genetics problems is to pick clear symbols to represent the traits, and then to set up the Punnett square to predict the outcomes in the offspring. Since women have two X chromosomes, cells balance the activity of the X chromosome by randomly inactivating one of the X chromosomes in each cell during early stages of embryo development. From the standpoint of the whole body, both X chromosomes are active, but each cell uses only one or the other X chromosome. How does this happen? Watch the animation on X chromosome inactivation, available as a link for this unit. The inactive X chromosome is called a bar body and can be seen as in high magnification as a tiny dot in the cell's nucleus. This diagram shows the effects of X inactivation on the calico cat. The gene for fur color is located on the X chromosome. Cats have two different color alleles, one for black fur color and one for orange fur color. If female cats are homozygous, they'll either have black fur or orange fur. However, if they're heterozygous, the inactivation of the X chromosome in the embryo produces patches of cells. Some have an active X chromosome with black fur color, and some have an active X chromosome with orange fur color. The result is a mosaic of cells that gives the overall calico phenotype. For our last topic in this episode, let's consider the consequences of chromosomal changes. One type of chromosomal change can occur in meiosis, in which chromosomes fail to separate during the anaphase stage of either meiosis I or meiosis II. In either situation, the resulting gametes will not have the proper number of chromosomes. This type of chromosomal abnormality is called non-disjunction, and it results in a condition called aneuploidy. One of the most commonly occurring forms of non-disjunction in humans is Down syndrome. Down syndrome occurs when an extra chromosome 21 is passed in either the egg or sperm. The extra copy of chromosome 21 affects both fetal and child physical and mental development. This condition is also called trisomy 21, referring to the 3 copies of chromosome 21. The incidence of Down syndrome is correlated with maternal age, and its frequency is nearly 1% of births in mothers over age 40. The reason for the increased incidence in older mothers is not completely known, but it may have to do with the increased age of the eggs in older women. There are several other types of chromosomal trisomies, but these often result in miscarriage or in very low infant survival. There are several other types of chromosomal disorders that have been noted and studied. Klinefelter syndrome occurs when an individual inherits an extra X chromosome from either the sperm or egg. And Turner syndrome is a condition in which an individual has a single X chromosome. Since the Y chromosome determines maleness, individuals with Klinefelter syndrome are males, and those with Turner syndrome are females. Both Klinefelter and Turner syndrome individuals develop fairly normally, although they may be sterile. So why does having an extra chromosome 21, as seen in Down syndrome, cause so much trouble in development, whereas the extra X or missing X not seem to affect physical or mental development as much? The reason lies in X inactivation. Any individual has only one active X chromosome per cell, so Klinefelter individual with their XXY genotype will inactivate one chromosome per cell and end up with a normal chromosomal makeup. Similarly, Turner syndrome individuals are XO, but since only one X chromosome is needed per cell, they have the normal number of active X chromosomes. Another category of chromosomal abnormality is alteration in the structure of a chromosome. In some instances, pieces of chromosomes are deleted or duplicated. This may have drastic effect on an individual, particularly if the deleted regions carries essential genes. Chromosomes may also have regions that are moved by inversion or translocation. One type of cancer, chronic myelogenous leukemia, is associated with a chromosomal translocation. You should now be able to describe the chromosomal theory of inheritance. You should be able to explain the inheritance of sex-linked traits and be able to explain why sex-linked diseases are more common in human males than in females. You should be able to explain the terms X inactivation, non-disjunction, aneuploidy, trisomy, and translocation. And you should be able to answer questions such as page 289, 1 and 2, page 292, number 1 and 2, page 303, number 1, 3, and 8, and you should be able to work the problems and questions on the MyLabs Plus homework for Chapter 15. This concludes this section of BioB 160 Principles of Living Systems. Be sure to read the chapter and post your questions and answers to this week's discussion board. Thanks and have a great day.